Thank you so much. I'm so excited to be here today to talk about science. Has everybody had their mind and energy to get through a science presentation? Great, great. Here at ASEA, we have a vision to become the global leaders in redox and cellular health. With that vision comes responsibility. Anytime you assume the role of leader, there is a responsibility to lead from the front. So with our vision comes the responsibility of performing research in the area of redox, educating on its benefits and raising awareness about the life-changing opportunities that come with redox, and continuing to build products that leverage the technology to change people's lives. Now, earlier this year, in an effort to raise awareness and to help people understand how redox works and what are the benefits that to be had from redox, we released some key words. These key words are an effort to boil down what are complicated topics and make them easy to understand. So today, in addition to talking about some research that we're extremely excited about to share with you today, we're going to talk about some foundational principles about redox. But before we go into the keywords and how to be able to understand these, we need to understand redox as a whole at a foundational level. So first, redox. What is redox? Redox is simply a contraction of two words, reduction and oxidation. And I love this quote. This quote is extremely profound and will be important to our discussion later. Life is nothing but electrons looking for a place to rest. Remember that, looking for a place to rest. But what is an electron? An electron is a very small negatively charged particle. These particles have a ton of energy associated with them. And the concept of electron transfer, which is central to redox, is the process by which an electron is transferred from one species to another. Redox is the transfer of electrons, and the transfer of electrons is the transfer of energy. You cannot achieve cell signaling without energy. Now remember, looking for a place to rest. I want to go through this simple demonstration, this simple example to illustrate what I mean, looking for a place to rest. Imagine a ball on top of a hill. What happens if I give it a nudge? We all know intuitively that the ball rolls down the hill. When I go through this experiment with people in this thought experiment, I say, hey, so why did the ball roll down the hill? And I almost always receive the answer, well, gravity. We all know that gravity pulls the ball down the hill. And then I ask the question, well, why does gravity pull the ball down the hill rather than push it up the hill? What is gravity doing? What is gravity trying to take from the ball? And the answer is energy. I heard it. At the top of the hill, the ball has potential energy. But what did we hear in that quote? Looking for a place to rest. Everything in this universe is looking for a place to rest. The ball contains energy at the top of the hill that it does not at the bottom. So when nudged, it will spontaneously roll down the hill looking for a place to rest. So now let's apply this to electrons. When we talk about redox, we talk about electron transfer. Why would an electron transfer? Well, if we think about the ball rolls down the hill to achieve a resting place. Much like the ball, electrons transfer to achieve a state of rest. In chemistry, we use the term oil rig. Oxidation is lose, reduction is gain. So when a species is oxidized, it is giving up an electron. When a species is reduced, it is gaining an electron. Now, why does that electron transfer from species A to species B? It's because there's a lower energy level available in that second species. That electron is looking for a place to rest. And when it transfers, it releases its energy. So now that we understand why electrons transfer and that energy is released upon a redox transfer, let's talk about how do we make redox signaling molecules. Redox signaling molecules are tiny, high-energy molecules. To make these molecules, we use a patented process to create these high-energy molecules. We start at the bottom curve there with sodium chloride, salt. Salt is relatively low energy, and when we dissolve it in water, it goes down the energy curve. You can see the ball rolling down the hill to a low-energy saline. Now, with that said, the question is why does salt dissolve in water? The answer is salt is at a lower energy level dissolved in water than as a solid. Salt is looking for a place to rest. Now, in order to achieve cell signaling, energy is required. Salt does not have enough, saline doesn't have enough, so we have to introduce energy into the system. So with our patented process, we push the ball back up the hill, so to speak. But we don't want to go back up to the same hill, we want to go to a different hill, a hill of higher energy, the hill of signaling molecules. Now there on the left of the graph there, you can see we have energy on the y-axis there. In redox chemistry, energy is called redox potential. So when we say at ASEA, we power potential, we do not simply mean your potential as an individual, your potential to change the lives of those around you, and the potential of your business. We mean very literally, we power the potential of your cells to signal and communicate. Okay, so we're going to dive into these signaling molecule keywords because they're going to pop up in the research that we're going to present here shortly. I like to go through the keywords very succinctly so that everybody understands how they're related. Cell signaling molecules are tiny, high-energy molecules. Because they are tiny, they can be absorbed directly into the cell. Once they're absorbed, they act as an on-off switch, turning on signaling within the cell. Once that cell is on, it signals to the cells around it. Those cells signal to the cells around them, beginning a cascade or domino effect. And it's this cascade or domino effect that contributes to our resilience to stress and overall wellness. Okay, so we talked about tiny molecules. Now, what we want to do is we want to say, okay, because they're tiny, they can be absorbed, right? We've done studies at the corporate office, at our labs, and we have found that absorption happens very rapidly with signaling molecules because they are tiny. We have found that the majority of the signaling molecules are absorbed within the first 15 to 30 seconds in the mouth. That's why we recommend that you swish it before you drink. Once you swallow, the absorption continues throughout the body. But what does it mean to be absorbed? Well, in the case of signaling molecules, they undergo what's called passive absorption, meaning because they're so tiny, they're free to pass through the cell membrane without permission. They can be readily accessible to the cell and absorbed so they can go straight to work. Okay, so signaling molecules are tiny molecules that are absorbed into the cell. Once they're absorbed, it flips the switch on in the cell. Once that cell is turned on, it begins to communicate with the cells around it. Those cells begin to communicate with the cells around them. And that begins the cascade or domino effect. You can see here as that signal spreads throughout the cellular network. And it's this cell signaling cascade, this domino effect, that contributes to our overall health and wellness, our resilience to stress. Now, this concept of resilience to stress is extremely important. And I want to pause here because this is an important concept. Your overall health and wellness, your ability to maintain your health, is almost entirely dependent on your ability to respond to stress. Let me say that again. Your ability to maintain your health is almost entirely dependent on your ability to respond to stress. So let's imagine I'm in the middle of a sawhorse here, trying to do a balancing act. What happens is I'm exposed to stress and knocked off center, knocked off balance, knocked off homeostasis. Again, central to my ability to maintain my health and well-being is my ability to respond to that stress. Redox signaling molecules enable us to respond to stress and get back to center. So as a quick review, before we move into the research, tiny molecules are high energy molecules. Signaling molecules are tiny high energy molecules. Because they are tiny, they can be absorbed into the cell. Once absorbed, they act as an on-off switch, turning on cell signaling. Once a cell is turned on, it signals to the cells around it. Those cells signal to the cells around them, beginning the cascade or domino effect that contributes to our resilience to stress and overall wellness. Now again, at the beginning of this presentation, I talked about our vision. We take this responsibility of being the global leaders in cellular health and redox-based technologies extremely seriously. We invest heavily in product development and research. We know this product can change lives all around the world as it has already done so. So this year, we've been conducting research with partners around the world, at universities around the world. And we want to share some of those results today. And to help me do that, I'd like to introduce Dr. Andrea Borges. You need this? You need that?
Thank you, Hunter. Good morning, everyone. You ready for some science? Yeah. Okay, I hope you took your energy and your mind this morning, because I really need your focus, a little bit of your energy, and a lot of memory. So, thank you, Hunter, for the introduction, and I would really like to express my gratitude for this amazing opportunity. It's a pleasure to be here to share some very exciting news that we obtained with our research, and I want to share this with all of you today. Well, over the past months, our team has been working very hard on different projects. We want to understand better the redox signaling molecules and their effects in our body and in our cells. So it has been a journey, and we know that science is very complicated. So it's been a journey of dedication, collaboration, and a lot of hours of very hard work. And I'm very glad to report today some of the results that we obtained and that hold very important and significant good implications for our company. So before that, I would like to go back to 2017, when the Tarad Lab performed a very nice and very important test with the ASEA Redox, our blue bottle. Let's talk a little bit and very briefly about this study and how it connects to our current projects nowadays. So a quick recall of this study: it was an 8-week double-blind placebo-controlled human trial. There we go again with those big names, right? But just for you to know, you don't have to remember anything like that, but just for you to know, this is a big name, just to say that it's a valid and very robust method, and that validates our test and the effectiveness of a compound or an ingredient or a chemical, and in our case, ARS, or the ASEA Redox. TARED study showed that several genes were tied to key functions and very important pathways in the cells. And few of these genes were upregulated, meaning that the cells were making more of these proteins because the genes were turned on, like Hunter just mentioned, right? Genes were turned on in the group of subjects that took ASEA Redox. So among these genes, there were 5 genes that were turned on. These very complicated names, right? But in the left column, we can see the names of these genes. And in the other column, we can see how much the gene expression increased compared to the group that did not receive ASEA Redox. Even though they're complicated names, all we need to know is that they are very important because each one of these genes is related to different pathways that will produce specific proteins in our cells. The TARED study showed that ASEA Redox impacted the modulation of 5 different genes. These genes are related to many pathways, and here we listed 15 important pathways that these genes are related to. So inside each of these blue squares, we can see the name of the pathways that these genes are related to. They're all very important for our overall health and resilience. I know they're very complicated names, but looking at the pathways, we decided to go deep to understand some of them. And the first one that we chose and that we want to understand better, it's an important pathway that is totally related to oxidative stress, which is the NRF2 pathway. So ASEA partnered with universities around the world to find out the relation between redox signaling molecules and NRF2. Once again, I'd like to highlight that these are all very crucial pathways for cellular function. They're all very important for cellular health and, again, for cellular function. So in order to start to understand the ASEA Redox mechanism of action and the function of redox in our body, we narrowed down the studies to NRF2. But why did we choose NRF2? Why, among all of these pathways, these 15 pathways, why did we choose NRF2? Well, first of all, because we learned that NRF2 pathway is responsible for turning on and off genes that are very essential to produce protective proteins that will combat harmful compounds in our body. Remember that Hunter mentioned switching on and off? So that's what NRF2 does, is it turns on and off important genes. And this pathway also functions like a master switch, you know, the light master switch that turns lights on and off. So that's how we compare this pathway. We also understand that NRF2 is a crucial regulator of oxidative response. It promotes cellular resilience to stress. Another thing that Hunter also mentioned before, it promotes cellular resilience to stress. So our cells are able to be challenged. What does that mean, resilience to stress, right? It means that our cells are able to be challenged by insults and respond and restore their functions just because the stimulation of NRF2 pathway brings back the balance of the cell. So remember this, resilience to stress. That's what NRF2 is related to. And finally, because genes that respond to NRF2 pathway they are involved in protection of the cells against oxidative stress. Well, actually, the NRF2 is one of the major pathways in our body that is involved in the adaptation to stress. When we activate, when we turn on NRF2 pathway, we create more antioxidant proteins in a very natural way. And this is really important because it's physiological, it's how our body works. And this is really good because it allows our body to fight oxidative stress. So, in order to study and move forward and understand the research on NRF2, ASEA partnered with 2 different important universities around the world. Currently, we are doing research with University of Bath in the UK to determine if Renu28 modulates NRF2, and Western Sydney University in Australia. We partner with them to investigate the role of ASEA Redox in the activation of NRF2. So far, we obtained very interesting results regarding the activation of NRF2 by Renu28 and by Redox, by the ASEA Redox. And I would like to dive a little bit more into the science of these 2 universities and the results that we obtained from them. And with that, I would like to start with the University of Bath. University of Bath tested Renu28 in skin cell cultures, and the scientists from Bath divided the experiments in 2 groups. If you're in the science behind redox on Wednesday, we talked about it. It's very important to have 2 different groups in the tests, in the experiments. One of the groups is the control group. This control group never receives any kind of treatment. The other group is the group that will receive the treatment. In this case, the cells were treated with Renu28. So with these 2 different groups, they performed 2 different experiments. So in each experiment, there were 2 groups. The first experiment, we wanted to evaluate Renu28 if Renu28 increased the production of glutathione. Glutathione is one of those protective proteins that we just talked about that NRF2 can modulate. So Renu28 was added to skin cell cultures, and the amount of glutathione produced by these cells was measured. So we observed there that when Renu28 was added to the skin cultures, glutathione production increased by 40% compared to the control that did not receive Renu28. And I want to highlight here, and remind you that glutathione is a protective protein, okay? In the second experiment, and I actually love this experiment and I love this result, it shows something very unique. And it was really a surprise even to me. These results are represented in this graph that it just came up. And it said that the glutathione production was blocked with a very specific glutathione blocker. Okay? That means that the production of glutathione was not happening in those cells. Then, when they realized and they were able to measure a 0 production of glutathione, they added Renu28 to this cells that were not producing glutathione. What was the result? The result, this is what happened. When they added Renu28, the cells were able to restore glutathione production by 55%. So what does that mean? When our body is under stress, these protective proteins can be compromised. The production of them is compromised. And we're always under stress, right? What we saw here, these results might suggest that Renew28 can restore the production of glutathione. Glutathione, again, is a protective protein in the skin when the skin is under stressful conditions. So, now that we know and we got to know that Renew28 increased glutathione levels, the next step was to understand if Renew28 was activating NRF2 pathway. Because glutathione was there, how was the relationship between glutathione and the cells and Renew28 and NRF2? Remember that NRF2 pathway, again, is the master switch to turn on and off genes that will produce protective proteins. And glutathione, again, is one of those protective proteins. So this part of the study, different dyes were used because they provide color to the experiment to facilitate and to facilitate the understanding of the results. So let's understand these pictures, these images, and these colors. The upper pictures, the upper images are related to the tests with skin cells, and these skin cells did not receive Renew28. The bottom images are related to the tests where Renew28 were added to the cells. So the skin cells received Renew28. So after the treatment, the cells were analyzed for the presence of NRF2. Let's understand the colors now, what blue and what green means. The pure green images show that there is NRF2 inside the cells. Everything that is green is NRF2. The blue color is just the nucleus of the cell. So in the last column, there's an overlap with those two images. So we overlapped green and blue, and we create new images with new colors because these two colors were mixed. And this will help us to understand where things are happening inside the cells. In this case, we know that Renew28 activated NRF2, and we also know that NRF2 is working in the nucleus because the original blue color of the nucleus mixed with the green color of NRF2 and turned into a kind of aqua blue when we overlap both of them. And this is happening only in the bottom image, where the cells were treated with Renew28. So with that, we can understand and we can observe that NRF2 can only stimulate gene expression when it's in the nucleus. And this result means that NRF2 is there in the nucleus regulating gene expression, turning genes on and off. And taking together both of these results from Beth, the first one that we showed and this one, we can conclude that those are very, very amazing findings that suggest that Renew28 promote the production of protective proteins. In this case, glutathione. And we could also conclude that this production happens through the NRF2 pathway. Another study that we performed now with ASEA Redox was performed in Australia by the Western Sydney University. It was also an in vitro study using cultured cells to study the effects of ASEA Redox on NRF2. So cells were again cultivated in the presence or not of ASEA Redox. This study is very important because it showed us that ASEA Redox, the blue bottle, increased NRF2 activation in about 160%. So what does that mean? It means that genes responsible to produce protective proteins, and some of them are listed there, and you have to memorize all of them. That's why we need a lot of memory, just kidding. Okay, so those protective proteins can be regulated and those genes can be turned on and off to start to work. When they're turned on, they start to work and produce these protective proteins. These are some of the antioxidant genes that can be regulated, that can be turned on by NRF2. It also means that the antioxidant response system is on in the cells. It means ASEA Redox turned the switch on in a very natural way. And turning on a switch in a very natural way is very important to our body, because our body is already under stress. We don't want more stress if it's not physiological, if it's not natural. And ASEA Redox promotes that, promotes this genes to be turned on and off in a very natural way. So what we can conclude from both Bath and Western Sydney studies? First, we can conclude that ASEA Redox as well as Renu28, meaning redox signaling molecules, right, found in these products activate NRF2. Also, that redox signaling molecules in ASEA Redox and in Renu28 activated the master switch regulator, that's NRF2, to fight oxidative stress. And finally, this activation will lead to the production of those protective proteins. In this case, in the case of the study that we conducted in Bath, it was glutathione. So this antioxidant and these protective proteins, they are part of the antioxidant system, and it's very, very important to the cells because this is the system that will allow the cells to adjust and respond better to stress. It will help our cells to develop processes to be more resilient, and to adapt to stress. Remember one of the words of the 5 keywords in the redox signaling. So it will promote overall well-being, which matches directly to our vision, which is to become the recognized global leader in cellular health and redox-based technologies. So